Hello, this is the CUDA and Open Lab seminar, Cultural Data Analytics, on March 9, 2026. And today it's our greatest pleasure to have Elisabeth Bik, who is very well known for the work she will present today. It's about errors and misconduct in biomedical research images. And those of you who may wonder why we invite Elisabeth Bik to Cultural Data Analytics, there's two reasons. One is I'm familiar with her work since quite a while because I basically did my PhD on the visual transmission and building reconstruction over the last 650 years, which has a lot to do with visual family resemblance as Wittgenstein and later Eleanor Roche defined it. And there's very few art historians, surprisingly, or relatively few, who actually still work with this image similarity concept, where this is the central thing. And in that sense, I think Elisabeth Bik is one of the most prominent art historians, in quotation marks, and we can learn a lot from her. And the other thing is that systems biology, multidisciplinary science more general, are certainly cultural systems. And today we get a look into that in a way which probably the field didn't anticipate before Elisabeth Bik started to do her work systematically, which is super, super mesmerizing to look at, because if it wouldn't be that depressing, it would actually be fun. With that, dear Elisabeth, the stage is yours.
Thank you so much. No, my pleasure. Good morning from near San Francisco, where it's 7 a.m., so early in the morning. Still dark outside. You hopefully will have a sunrise in a bit. So my work is about error and misconduct in biomedical research images. I hope you can see my screen. This is all good? Looking?
Yes.
Okay, great. So I am, just a little bit about myself. I'm originally trained, did my PhD in the Netherlands. I'm a microbiologist. I worked on cholera, I worked on tuberculosis, all nasty diseases. I worked in a hospital. And then I moved to the US, where I joined the lab of David Relman and worked in microbiology. Again, specifically on the microbiome of humans and dolphins. A weird combination, but that's how it goes, I guess, in research. During that time, I started to become interested in science misconduct. I'll talk about that in the next slide. I worked also 2 years in industry, one of which was one of these jobs I had was at a company that later got raided by the FBI. So an interesting story there. I already had left the company, but that's not really relevant for my work. Now I'm an independent scientist. I quit my job to devote my time fully on science misconduct. I'm still affiliated with Stanford. I have a sponsored affiliateship, which means I don't earn any money. I don't have an official position at Stanford, but I have access to the library, which means I online, with my Stanford credentials, can actually scan the biomedical literature, because there's still a lot of papers, of course, behind paywalls. You can find me on social media. You can see that all on my slides. But because I'm independent and I'm not employed, people are always interested in how I make my money. So my financial disclosures are that sometimes I get money to give a talk, or to visit university and present my work, or to give a keynote at a conference. So I sometimes get speaker fees for that. I might also do consulting work for publishers or institutions that have received allegations of misconduct and who need an opinion about whether or not images or plagiarism is at play. And I also receive donations through Patreon. So Patreon is a website where people can donate a small monthly fee, and together that helps me give me a basic income to do my groceries, I guess. I'm not making a lot of money, but I'm very grateful for people who support my work, because this type of work, basically looking at the literature and scanning for troublesome papers, is not really work where you can normally get a grant for. I also have free access to a couple of software tools that I'll discuss later to help me find these duplications. and to the Stanford Library, as I already mentioned. So how did I start my work? I was working at Stanford on the microbiome of humans at that time, and I heard about plagiarism, and I don't even remember, I just was reading about it, and I just thought, maybe somebody had plagiarized my work. Let's check that. So I just took a sentence from a review article that I had written a couple of years prior and put that sentence into Google Scholar. And lo and behold, I have chosen a sentence that somebody else indeed had plagiarized. Somebody had copied my work. And what I found was this online book chapter in Intech Open Book Chapter, which is a very fishy publisher, but that's not really relevant for the story. It was just like a book, and one of the chapters you can see, it's a very short chapter. You can see the text here on the left of your screen. And you can see that I've marked this text with colors. And I'm not sure if you can see my cursor. Can you see that?
Yes.
Or? Okay, great. So you can see this text here in red. That is my text, actually. They had copy pasted my 2 paragraphs that I had written and, didn't even cite my work. I was not in the references. And so you can also see text marked in other colors, in pink and green. They had also stolen a lot of text from other people. And so this was this patchwork of plagiarism. And it made me very mad because somebody had stolen my work. And I started working on plagiarism. It was almost like a hairball. I searched for a sentence that I had written. I found this particular one. It had sentences stolen from other papers. And when you search for these sentences, just in Google Scholar, between quotes, let's say 10 words or so, I found more papers that had stolen that work. And so it was almost like a hairball. You sort of find one sentence, you find more. And I've kept on finding papers and papers that had plagiarized other people's work. I did that for about, a couple of months and found about 80 plagiarized articles, PhD theses, etc. But then I found a PhD thesis, and you can see some of the images from that PhD thesis in the middle and on the right on my screen. I found duplicated images. And this PhD thesis had plagiarized text, which is how I found it, but I was looking at these images of Western blots. Now, I realize this is a very wide audience. Not everybody might be familiar with what Western blots are. I'm not going to explain the exact details, but it's a technique to look at the expression of certain proteins. But for this particular slide, you only need to know you see these horizontal dark stripes on a light background. And I found several figures in this PhD thesis that had these Western blots. And the one that caught my eye was the one here that I see here in the bottom of the middle, the center panel. You see these 4 horizontal stripes and a little dot here. Now that dot that caught my eye. It's just it doesn't really. It's more like a little spot or stain. It doesn't really have any meaning in the essay. But I found the same spot, little spot here, in another photo, in another chapter of this PhD thesis, where it represented a different experiment. If you look at the labels, this is a time series, and here that same figure is something else. Like they were adding different compounds to see how the protein expression would change. But more importantly, you see these same four bands with this little dot, but it is in mirror image. So it's the same, that little dot gives it away. So I can see that these bands all have like a certain shape. They sort of curve a little bit upwards. We call it smiling. And all these bands have slightly different shapes. So I can see it's the same photo. And not everybody is very good in that, but I think I have some talent. It's the same photo, it's a mirror image, and it represents a different experiment. And not only that, I found a third occurrence here of that same dot with our little spot here. And again, a different experiment. And then I found this other set of duplicated photos that you can see here marked in red. So these little colored boxes are my addition. They're not part of the original figure. They're my addition to show that these images appear to be the same. And and again, this seemed in combination with the plagiarism that I found in the introduction of this PhD thesis, this seemed to be intentionally done, because if you flip a photo, that doesn't happen by itself, I guess. And these chapters had also been published as scientific papers. So I reported those to the editor, and both got retracted. Took a while, but they did get retracted in the end. So I realized a couple of things. I was just like blown away, like, why would anybody do this? And why did nobody notice this? So I'm very good in spotting duplications. If I go to a bathroom in a restaurant, or to a hotel room, or even in my own home where I have porcelain tiles, I like to look at patterns. And maybe some of you are crazy like me, or maybe somebody has never realized that sometimes you can see repeats in these patterns. Of course, when it's a real wooden floor or a real natural stone tile, you wouldn't expect to see these patterns, because in nature they're always slightly different. We don't see the exact same patterns. But in laminate floors or porcelain tiles, which are not natural stone or wood, you might see these patterns. And so I love to look at photos of these floors and like see, oh, this plank is the same as that plank, or this tile is the same as that tile. And I've always assumed that everybody does this, but I guess not. I'm pretty unique in this. Although I have occasionally people on Twitter will send me a photo like, oh, we found one as well. And thank you for opening our eyes to these, fun activity in your hotel room or the restaurant bathroom. But more importantly, I also realized, of course, what made me mad about these duplicated photos is that for me, it's a deviation of the truth. When we do experiments and we publish about that, we should be reporting the truth. For me, science is about finding the truth. That is my core belief of what science should be. This is perhaps different for everybody else, but for me, that is what science should be about. We do our experiments, we report on them, and we report honestly on what we found. We don't try to hide things, and we don't try to change things and make it look better. But also, science builds upon science. Science papers do not stand by themselves. We build upon the work that others have done before us. We build upon decades or even centuries of research that other people have done that helped us do our research. We cite other papers that cite more papers, that cite even more papers, and other people will cite, hopefully, our work. Our scientific papers are like bricks in a wall upon we layer new generations of bricks. Every paper rests on a lot of other papers. But if some of those papers, some of those bricks are not good, if they contain errors or misconduct, even fraud, that means that part of that scientific wall could tumble down, that a hypothesis won't work. And so traditionally, when we peer review papers or when we read them, we assume that papers contain the truth. We tend to trust papers when we read them. If you peer review a paper, you might assume that those patients really existed, that data is really as it is reported in the paper. But unfortunately, like in any other field, science is not immune to errors or fraud. And of course, there's also this pressure to publish that is increasingly pressuring us to publish. Us, when I say us, I don't feel the pressure to publish because I quit my job and I don't have a boss and I don't need to care about that. But I'm in a luxury position that I cannot do that. But most people, whether you're a grad student or a postdoc or a professor, you'll feel from above this pressure to publish. This is what scientists are held accountable for. This is what determines how we're being viewed by other people. We love to look at these metrics, like how many papers did you publish? How many times were they cited? What was the impact factor? And when you judge a person's CV, resume, when it's a scientist, when you're hiring, let's say, a new faculty or you're hiring a postdoc, we love to look at these metrics and we love to make judgments about a person's value as a scientist just by looking at these metrics. So we love to look at these papers and we assume that they're true, but because people are sort of almost pressured to publish, there's this, some of these people will perhaps turn to misconduct, not everybody. But so just first a little definition about what misconduct is, which of course is different per country. In the US, it is one of three things. So science misconduct always assumes an intention to mislead, and it can be plagiarism, falsification, or fabrication. So plagiarism is where a person copies somebody's text or ideas without giving credit, like that one paper that I found that had plagiarized my work. Science misconduct can also be falsification, where a person obtains results but changes them, like change one value, make it a little bit higher so that that patient, instead of being a negative, becomes a positive for a certain test, or copy paste some more value so it looks like you had more data, or yeah, just make your data look better by changing them. Fabrication is making up data. It's not even doing an experiment, just typing in some numbers in a spreadsheet, drawing some beautiful graph without actually doing the experiment. Now, these three boxes are, very clear, and it looks like it's very easy. Even I don't always know if something is a falsification or a fabrication. I feel these definitions are not always very clear, but this is sort of what the US government at least defines as science plagiarism, as science misconduct. But for example, that photo that I found that had been duplicated, is that falsification or fabrication? I don't even know that, because the data, one experiment did not happen, but the other did, and so this is a technical discussion. But there's also many things that I feel are also science misconduct, but that are not falling under this definition. So I don't think the definition is very good, but this is sort of what we have to deal with. Now, I do want to stress there's an intention to mislead, because people can make errors. People can choose the wrong statistical method, whether on purpose or not. People could leave out negative results. So those practices are usually called questionable research practices, QRPs. And again, a slide like this assumes it's very clear when something is a questionable research practice or science misconduct. I don't find it always very clear. A lot of authors will tell you when you catch them with a photoshopped image, they will say, oh, oopsie, we made an error. I'm like, really? So if you say it's an error, then it's suddenly a questionable research practice. Yeah, it's sloppy. Sorry, we should have grabbed another picture, another photo. But I feel it's misconduct. That intention to mislead is really hard to prove. So these definitions are not always very practical, but this is what we have. Now, I already told a little bit about this pressure to publish. And this is just increasing. I mean, there's even institutions that are pushing their researchers to publish more and more, because the university will end up higher in the ranking in their country. So here are a couple of scenarios that I feel could lead people to do misconduct. There are individual fraudsters, of course, and there are almost like industrial scale type of misconduct. The individual fraudster, one particular scenario that I think is fairly common, at least in the US, I cannot really speak for other countries, is that of a person on a visa, like a postdoc or a grad student who works in the US, in my example, and who is on a visa. And if you are on a visa in the US and you lose your job, if you get fired, your visa immediately ends, and it means you have to go back to your home country. And I've been in that situation because when I moved to the US, I've worked on a visa, and then I got a green card, and then I got citizenship. But, those first couple of years are scary. If you are fired for anything, you have to go back to your home country, because no longer do you have a sponsorship. Now, if that poor graduate student works for a very demanding or even bullying professor who who is convinced that a certain hypothesis is true, and the poor graduate student does their experiment and they don't work or they don't have the results that the professor wanted, and the professor now has a meeting with the grad student or the postdoc and says, why don't your experiments work? Why do you keep on having negative results? I am convinced that the results should look like that, and they don't work with you. Maybe I should hire another postdoc, and they can do the work better. And maybe I should give the project to one of your coworkers. And this poor graduate student now feels a threat that they could be fired unless they produce the results that the professor wanted. So I think in this situation, a person might be tempted to Photoshop to make the results look like they are true. And I think this is fairly common with this, very competitive atmosphere in certain labs. So this is a common scenario that could lead people to do misconduct. Now, of course, there could also be people working in industry where you earn a lot of money, and I've been in that situation too, so I understand it. And you're sort of pushed from above to make the results look a little bit better. Maybe you work for a cigarette company, just to give a very extreme example. And your job is to publish papers or research that makes it look like smoking is really good for you. Of course, we know smoking is bad for you, but you can sort of see that there's an imbalance or like conflicting messages that you might need to send. Or maybe you are a person who loves to spread misinformation on their Substack, their blog, where they can make a lot of money because they have subscribers. And they have found that if they tell you that COVID vaccines are really bad for you, they make more money. And so there's a lot of people spreading misinformation about vaccines, and we see that. This pushing a false narrative. And then a third example would be people who work in a country where there's not a lot of money to do research, and where they're hoping to do a PhD or postdoc in a country where there is, some more money. And this is currently, of course, not the US, but the situation was, of course, that a lot of people from, let's say, countries like maybe China or Pakistan or Iran or India, they don't have a lot of money to do research. They would love to do a postdoc because it's a good booster of their resume, but they don't really have, they didn't have anything published yet. And so they might see advertisements where they're going to be an author, where they get help in publishing a paper. And these are advertisements by paper mills. I'll talk about that in the next slide a little bit more. These are people who want to have a couple of papers on their resume, and they don't have any ways of writing these papers themselves. So they fall for these scam paper mills. So here's a slide about paper mills. These are entities that sell authorships. They publish or they write. We don't really know how they work. They have different ways of doing this. But their main thing is that they sell authorships. So they will offer, you can go on Facebook, for example, you see these advertisements. They're called Call for Co-Authorship. Do you want to become an author on a paper? Send us some money and we'll put your name on a paper. That is basically what paper mills do. And they have different strategies. For example, an older strategy was that they worked with editors. They had a paper where it was already accepted, and then they will add a couple of extra authors on it. Or they could work with a template. We've seen this in cancer research, where they made these combinations of non-coding RNAs that inhibit the growth of cancer cells. And if you have a different non-coding RNA molecule and a different cancer, you can imagine there's a lot of possibilities. So you can sort of write the same paper over and over again, avoiding plagiarism detection, but sort of working from a template and changing a little bit and just reusing images from one paper to another. They just generated a bunch of images and then they use them for different papers. And because we now have software, we can actually see, oh, but that image was used in that paper. And of course, they have then probably a different or the same source. They could plagiarize or synonymize papers. We've seen that strategy where a lot of words, they basically the paper mill will take a paper or PhD thesis and they will change all the wording a little bit. They will plagiarize it, but without being detected. So they had special ways of rewriting the text, but some of these texts became very weird. I don't have an example for that, but we call those tortured phrases. Guillaume Cabenac was a person who worked on that. And he found that, for example, papers about artificial intelligence, instead of writing artificial intelligence, they would write counterfeit consciousness. And you're like, oh, what is that? Sort of, or like a paper about winged animals, and it was about birds. But the word birds was not in there. It was just winged animals. Or creepy crawlers were insects. Or I know, it's very funny. Or bosom malignancy was breast cancer. And so you can sort of synonymize a lot of expressions in that it's grammatically correct, but when you're reading it, you're just thinking like, what is this about? Like bosom malignancy, what is that? And so they Guillaume Cabenac built this whole how do you say, like a whole library of these expressions, and so you can find them, and found thousands and thousands of them. So that's a way that a paper mill might work. But of course, now we have artificial intelligence that can just in seconds rewrite the text without being detected by a plagiarism checker. So you can just take a random paper and rewrite it. So we see this influx of very low quality review papers that have already been published before, and somebody just rewrites it using AI in seconds, probably. So these paper mills continuously develop new strategies to avoid being detected. But it's estimated that the percentage of paper mill produced papers is enormously on the rise. So there's a paper by Reese Richardson. I have that here on the right, it published in PNAS, where this is a log scale. So, all scientific articles over time are on the rise, but paper mill products here in red are even more on the rise. Like, it's just enormously taking over. And this is just based on very smart ways of estimating whether or not a paper is a paper mill. So Reese, who published this paper, is part of a growing team of sleuths. I'm a sleuth as well. So I did not know the word sleuth. It means sort of detective in a funny way. We're post-publication peer review sleuths. So after a paper has been published, it went through peer review, and it was published, there are still potential problems with that, like the photoshopping or changing data. And some of those you might actually see. If you know what to look for, you could find these problems. So this is a growing group of people who do this. We all work in different ways. So I like to look at images. I'll show some examples. Other people look at plagiarism. I used to look at plagiarism, but I like images more, so I sort of switched. Other people look at statistical problems or clinical trials, or papers in cancer biology, or papers in material science or physics. There's, we all have different specialties and backgrounds, but most of us do this work either on the side or full-time, and we all have different styles of reporting it. Some of us work under our full name, like I do, and you can see some names there in italics, which are people working under pseudonyms, or groups of people working under pseudonyms. Because you can imagine that if you criticize somebody's work, that is not always received well. We are harassed. We are, sometimes sued. I have not been sued, but I was close to being sued. And so it's pretty risky work, criticizing other people's work. But these are some people I'm familiar with, I've worked with. I think this is a growing team. There's probably people I don't even know and I've never worked with who do this type of work. When we find concerns about a scientific paper, we post them on PubPeer. So pubpeer.com is a website where you can either go there and you can type in the name of an author or the name or the DOI, the unique identifier of a paper, and you might see that those people have their papers commented on. And you can also install a plugin, which is what I would recommend if you like to read science papers. So here on the left, I've installed the plugin on my computer, and this is a screenshot from a PubMed search. So PubMed is a library of biomedical literature. And if you've installed that plugin, you see these green banners appear when a paper has a PubPeer comment, and even you might see a red banner when a paper has been retracted or has received an expression of concern. So the plugin will continuously look at what you have on your browser, and if it sees the DOI of a paper that has a PubPeer comment, it will show these green banners. So especially for journalists or anybody doing literature searches on a regular basis, it works fairly well with PubMed. It works not directly in Google Scholar, but when you are on the landing page of a paper, so if you type in the DOI and that will bring you to the publisher's website, usually you will see these greenish or red banners appear. And then when you click on those banners, you will see the PubPeer comment. So this is a paper about SARS of patients in the intensive care, and I've written a comment about this paper, why I think it has a big problem. So you can comment on anything about the paper. You can give compliments. That actually rarely happens. It's almost like, negative reviews have a higher chance of showing up because people are mad about a product like a scientific paper, and they will write about something. Like, I think you cannot write this is fraud or this is misconduct. It's moderated. So you have to sort of keep a very objective and fact-based tone. But you can say, okay, this paper is missing a control group, or the control group is different than the treatment group at the start already of the experiment, so that's not good. Or this image looks remarkably similar to that image, but you cannot say this paper is a fraud or this paper contains fraudulent data. There's clear moderation guidelines, and so you have to sort of keep your emotions out of it. I'm out of moderation because I'm one of the power users, I guess. But I sometimes can squeeze in a slightly sarcastic comment, but even I get moderated sometimes. I focus on photos. Like I already said, I have this, I guess, talent for spotting duplications. But of course, if you think about scientific papers, there could be the data in those papers could take any form. Some papers might not even have any figures or tables. They might just have text or formulas, like math papers or philosophy papers. But more like in the biomedical sphere or material science, cancer research, things like that, the data typically will be either presented in tables, in line graphs, or in photos. Now, tables could have problems. You can sometimes see duplicated values, a particular standard deviation that occurs a little bit more often than you would expect. You can see that p-values don't match the average or standard deviation. So folks the sleuths who are more specialized in statistical problems or clinical trials might look more at tables. That is not what I'm very good at. Like, I can do some basic statistics, but I'm definitely not very good in interpreting ANOVAs or anything complex. So I usually stick with photos. But line graphs also offer sometimes some amazing results. And for example, so two of the line graphs here, these could be bar graphs, heat maps, ordination plots, things like that. Two of them at the top, these two are, or actually this one, this photo or this line graph is one that I made, so I know it's good. This is one of my dolphin project. But you can imagine that if you type in some numbers in a spreadsheet, if you're fabricating data, making up data, who knows, is this graph real or not? We assume it's real. And we assume, there's replicate experiments because we see an error bar here, but who knows? In theory, this data could be typed in a couple of seconds and the graph could be made, and maybe there was no experiment. And so it's really, really, it's much harder, I feel, to look at line graphs and spot problems. But occasionally we'll see things in plots like repetitive values, and I have an example of that later on. So, but if you think about photos, there's a lot of pixels. And there's a lot of unique things to look at. Now, I am talking about the time before artificial intelligence came in play, which was only 3 years ago. So most older papers, if you see a photo, we assume something happened. Like we say, a photo or didn't happen. A photo is sort of proof that an experiment happened. But also it has so many details. Like look at these photos of cells. You see these green fluorescent cells. And every photo is different. And we can look at that. We can either look by eye, like I did in the beginning, or we can use software to help us find overlapping images, because you wouldn't expect a different experiment to overlap with another experiment. This is a photo here on the right of a Western blot, or lots of Western blots, actually. And if you look at these bands, these horizontal lines, each of them has a slightly different shape. They might look like bones or fish or whatever you want to see in them. They might be smiling a little bit, or they might be thicker on one end, or they have little dots and specks in the background. So we can look at them, and there's an amazing amount of details. This is a photo here of a gel. And again, this is a DNA gel, maybe a PCR product or so. And you see all these little bands, they all have different shapes. Or photos of mice or plants or tissues. There's incredible amount of detail. So I focus on photos. That's my specialty. And of course, I cannot detect all the problems, but sometimes you can see problems in photos. What we can spot is not really photoshopping, but what most humans and software can spot are duplications. That is what our eyes can spot. We can spot different types of duplications. So here's an example here on the left of what I call a simple duplication. You see 4 different photos, and each photo has a different label. So it doesn't really matter what the experiment here is, but I hope we can understand that each of these photos should show a different photo. But 2 of these photos, and I've added these red boxes, 2 of these photos are identical. They're exactly the same photo. So this is a simple duplication. This could be an honest error. Like, this is not necessarily misconduct. And again, like I try not to point out fraud, but in some cases you have to think about the sloppiness that happened. But you can imagine that if you are a researcher and you took lots of photos of all your experiments, every experiment you put under a microscope, these fluorescent cells, you take photos, and maybe you didn't label one of the photos correctly, or you dragged and dropped the same photo twice. And so that can happen. So we assume these simple duplications are usually honest errors. But, it's worth pointing out because there should have been a different photo. And we don't know, is this photo the correct one or this? But one of them is not the correct one. We can safely assume that. Another type of duplication is a repositioned duplication. Again, in this example, we also see 4 photos, but the photos overlap with each other. Again, the photos, if you look at the labels, they should be different experiments. They're actually 2 different cell lines and 2 different treatments. And all the photos overlap with each other. Every photo came from the same sample. So basically what happened here is either a photo was cropped, one big photo was cropped in different ways, or the sample was moved under a microscope and different photos were taken. The photos should not overlap. They should all be not overlapping. And this could, I don't know, when you have 4 experiments, but all photos came from the same experiment, that is either extremely sloppy or there was an intention to mislead, as we usually say. And repositioning could also involve mirroring, like I've shown in that example, or rotation or stretching, things like that. On the right, there's just one photo, but the photo itself, that one photo has duplicated elements. You can see this same part of the tissue is also visible here. Now, that is not expected in a normal tissue, that you see these exact pixel level duplications. So half of this photo came from the other half of the photo, pretty much. So here we assume this was deliberately done. We assume that whether or not it's misconduct is different, but we assume those pixels didn't duplicate themselves. This photo was altered. This photo was changed with an intention to mislead, because presumably there was something in either this half of the photo or that half that the author didn't want us to see. And it could be something innocent. Sometimes it's just a scale bar or label, like the letter C or so. Sometimes it's even done by the publisher who didn't like C and said, no, it should be 3, or it should be a different font. And sometimes we see that these little corners are cloned. Corner cloning, we call that. But in this case, the whole image pretty much has been duplicated. And, you know, what was there? Was there like a hair or a scratch or like something blurry that the authors didn't want us to see? So we don't really know if it was innocent, but we know it was deliberately done. So these 3 types help us find or make at least some judgment on whether or not it was misconduct. But of course, to really know if something was misconduct, you need to compare the published photo to the photo that was obtained in the lab books. And for that, I cannot do that as a person just looking at papers. You know, when you report these papers, you hope that the institution will do an investigation. What were the actual results obtained and what was published, and why are they different? And so that could help a person investigate, like an institution investigation could try to figure out if it was misconduct, and also who has done it, which is a different question, and one that is much harder to answer. So I always love to play the game Spot the Duplication. I usually use that under the hashtag image forensics on either Blue Sky or Twitter. And I'm showing here a photo or a figure from a scientific paper. And you see 9 different photos, different labels. They should all be different photos, but 2 of these photos are identical. So this is a type 1 duplication. Again, we assume this was an honest error. I will leave this on so you can see if you have the talents. These are fairly easy to find. You know, for those who are online now, just raise your hand. I'm not going to call anybody out, but raise your hand if you've spotted it. Did you spot the duplication? Not everybody has. No, that's okay. But it's it's a fun game because, you know, it's, of course, it's an error. Again, I'm not accusing anybody of misconduct. We assume an honest error. This was actually a preprint in which I found it. And this was very quickly corrected by the authors. They said, oopsie, we made an error. This is what we want to see. It was corrected. And this was caught during, you know, before the paper was peer reviewed. So here are the duplications. These 2 panels are identical, and they should not be. There are different labels, there are different cell lines or whatever. I usually don't even understand what I'm looking at, but we see that the same photo is present twice. Here's an example of a type 2 duplication. So now we're looking at images that overlap. It's a simple one. I didn't put in a mirrored or rotated ones, which are much harder to find. And even the samples the photos that overlap are next to each other. So it shouldn't be too hard. Now, when you look at overlaps, we could sort of rule out panel A. Panel A looks very different than any of the other panels. So you sort of make these pairwise comparisons. Which of these panels could be overlapping? Now, this panel, panel D, also looks different. Maybe it could overlap with E, but I don't see anything. But if you look at panel C and F, they look fairly similar. And so those might be good candidates, just to help you, good candidates to find an overlap. And so these panels do overlap, indeed, C and F, and they're even in the same orientation. So this should be fairly easy to spot the overlap if I sort of point you towards the direction. And I do say, like, I used to find these things by eye. Now I have software to help me find it. And once you point it out, once I draw my little boxes around it, you probably will agree that these samples indeed overlap. if you look at the legend, they should not overlap. They're different samples. So this is an example. These are scanning electron microscopy photos of nanocomposites. This is material science. I don't really know much about material science, but these scanning electron microscopy photos should not overlap. These are the hardest, the type 3 duplications. So we are looking at two photos. They don't really overlap, but within each of these photos, and both photos came from the same paper, which within each of these photos, we might find duplicated areas, a type 3 duplication. Again, not saying this is misconduct, although I think of the three examples I'm showing, this is the most likely to have been the result of misconduct because there's heavy duplications in each of these photos. And it's maybe not easy to see unless I point it out. Like, for example, if you look at photo L, this structure, you also see partially here. Or, let's see, this little orange or reddish sort of diagonal. Like I like to look at diagonals, like things are on the same line. So you see that here and here and here. So once you start seeing these things, or maybe you see this little red cell with two blue blobs in it, you see that here too. So once you start to find these things and you annotate them, and this is actually, I love doing this. This is like, oh, a whole afternoon of me annotating this photo. And you find more and more and more. So the software will pick up some of this, and then you start looking by eye and just staring at it and like, oh, wait, I've seen that before. And finding all these duplicated areas. So this goes beyond corner cloning or covering up a hair. I don't know exactly how, of course, the original photo looked like, but these are extensive duplications. And so I've reported this, but nothing happened so far. So hopefully this paper will be retracted, but it's been on paper for a couple of years and nothing has happened so far. Sometimes we see these duplications not in photos, but also in line graphs. So, and once you see them, like I used to completely ignore these types of graphs because I didn't quite understand what EDX or XRD technology is, and I still don't quite know how it works. Again, I'm not a material scientist, but very often these are techniques used to test some particular chemical or physical properties of a particular compound. But I know that, for example, this graph on the right, you usually see noise and you see some peaks that have some meaning. Again, I don't really know what I'm talking about, but we don't expect the noise to be repetitive. And still, if you are paying attention, sometimes you might catch these repetitive noise patterns. Or here in this EDX panel, I've now learned, like Reese Richardson has helped me a lot in understanding these graphs, or at least understanding them to the point where I can catch a couple of problems. Like these are elemental peaks, and the elements should be at a particular position. Like C should always be, I don't know, around 200 or 300, and the O, the oxygen peak, should always be around 500. And so if you know these rules, sometimes you see the oxygen all the way over here or here, but these are all at the right position. But in this particular example, there's some noise here that seems to be repetitive. So maybe they were trying to brush away a peak that they didn't want you to see. So these are examples of material science papers with altered type 3 duplication. So I assume this is an intention to mislead, not just beautification, but we don't really know. So this type of work, looking at images, I've done now for 12 years. But when I started finding these examples, I wanted to know how often would you find these types of duplications in biomedical research publications, which are my home field. And I still was scanning by eye. I didn't use any software at that time to help me find these duplications. And because I'm slightly crazy and weird, I scanned 20,000 papers. I was still fully employed at Stanford. And I just wanted to know how often would you find that. First, I thought, okay, let's look at 100 papers. And then that's maybe not enough to have a good opinion about it. Let's look at one journal, or let's look at a couple more papers. Let's look at more journals. So in the end, it became 20,000 papers from 40 different journals and 14 different publishers. But about half of these papers that I scanned were from PLOS ONE. That's what I started with. And I added these other journals later. In that set of 20,000 papers that I scanned by eye, I found almost 800 papers that had duplicated images within the paper. Because I scan by eye, at least at that time, I don't have a perfect photographic memory. So I cannot really recognize that one image has been used or taken or stolen or reused from another paper. I can only look at that paper at that time and then sort of reset my brain and then go to the next paper. So 4% of those 20,000 papers contain duplications, inappropriate duplications, I should say, because sometimes the duplication is quite okay. And I had to learn when they were okay. And that's why I needed two co-authors, Arturo Casteval and Ferric Feng, who were both editors-in-chief of journals. So they were quite experienced, and they had also published a lot about science misconduct. And they had to agree with me. So I did the scanning, I found duplications, and both of them had to agree with me. If they didn't agree, we didn't include it in that set. And we agreed on 90% of the cases. In the beginning, I was a little bit too trigger happy. I called a lot of things duplications, maybe that weren't quite duplications or that were appropriate. And they were in the beginning not quite seeing the duplication. So we sort of grew towards a consensus over that year or two that we worked together. So 4%, 1 in 25 of papers that had a photo, at least one photo, contained these problems. And we estimated, and this is of course where we're not quite certain, that about half of them were done intentional. And that was when a photo was overlapping, rotated, stretched, mirrored, or when a photo had a type 3 duplication. We assumed it was intentionally done. Again, this is not the same as saying it was misconduct, but there was an intention to mislead. And then we got the question, like, does it mean that 2% of all papers contains potential misconduct? And that's, of course, a hard question because we looked at a specific subfraction of science papers. Again, papers that have photos. We didn't look at other fields like math or physics or chemistry. Excuse me. But I also know that it has to be an underestimate. Because imagine that if you have two photos that overlap and there's an overlap, I will spot that. But if you are a smart fraudster, you take a photo, you move your sample a little bit more under the microscope, a little bit farther to the left or to the right. There's not an overlap for me to find, and I would not be able to flag that. Or if you're a very good Photoshopper, you wouldn't just take one part of the same photo and copy, paste that, stamp that here and there. You would take maybe a band or a part of that tissue from another photo. There's many ways of cheating that would not leave any traces. And if you think about line graphs or tables, maybe it's even easier to cheat in those because, like I said, you could, in theory, somebody could type in some numbers in a spreadsheet and put it, make a graph without actually doing an experiment. So we think that the real percentage of misconduct has to be higher than 2%. We don't know what it is then, but maybe it's 5% or 10%. But I do want to point out, it's not that all science is flawed. Like, it's only a small subsection of science that could have these problems. And again, like, an intention to mislead is not necessarily the same as misconduct. It's really hard to prove misconduct because most authors will just say, oopsie, we made an error, even if it involves photoshopping. And then it's really hard to prove that they, committed a crime, science-wise speaking. Of that set of 800 papers that I found around 2015, I reported all of them to the journals, which is the correct way of doing it. You write to the editor-in-chief, hey, there's a problem in these papers. And then you check in periodically if those papers have been corrected or retracted or if nothing has been done. And it turns out in most cases, the journals are very slow to respond. So the 800 papers that I reported in 2015, around 2020, I looked at, how many of these have been retracted or corrected. And it turns out that 2/3 of these papers, 65.5%, there was no action taken. These papers were still out there with their error. And only 1/3 had either received a correction or expression of concern, barely visible here, a little sliver, or a retraction. So that is disappointing because 5 years seems like a good amount of time. You give everybody a good amount of time to address the issue. And now the number, so it was 2/3 no taken action. Now it's down to 40%. So these papers are still being acted on because I keep on posting them on Twitter or write a post about it, or I keep on writing to the editors like, hey, I sent this in 2015 and it's like now 10 years later and you still haven't done anything about it. So in some cases, these papers are still being acted on, but why does it take so long? That is a question I don't really understand. And it's frustrating. And this is why we post on Papier, because we want to warn other users. If a person is basing their new hypothesis, their new line of research on a paper that has a big error, I want to warn the world, like, maybe choose another paper. Don't build your whole PhD career on this particular paper. So we feel, it's nice that papers get retracted, but because it takes so long, and some papers can, some problems can be dealt with a correction, but we want to warn other people. So that is why we use Papier. Now, of course, other people will say, you're harassing these authors. And there are several authors who claim that we're harassing them because we found 40 papers of that particular author with problems. So I don't know, we're harassing them, or they did a bad job writing papers, but long discussions can be held about that. But my score, if you will, is that so far I've found over 9,000 problematic papers, most of those with image problems, but there's still like animal ethics, human ethics problems, plagiarism. It's there's different problems I found. I do focus on images, but there's some other problems in that set as well. Of those, about half of them I've reported to the journal's institutions. I'm still very much behind, but trying to work hard on that. But most of these papers I did post on Papier, so almost all of them. There's still some confidential projects. Sometimes I'm working on with, as a contractor or a consultant. So there's some papers I cannot post on Papier, and some don't have DOIs, or they're just fake papers, so you cannot really post on them. And my work so far has resulted in 1,600 retractions, 1,200 corrections, and a bunch of expressions of concerns. So I hope I've left some mark on science, but it's not good enough, I feel. So I want to switch gears. Are there any questions so far? I'm going to talk about AI a little bit in the next couple of slides.
Should we ask questions? I think it would be a great moment to actually take questions, because you
Sure.
sort of like are done with your basic technique.
Yeah.
I can ask a question until everybody else decides to raise hands. That's stemming from my PhD, which was focusing, as I said, on building reconstruction over 650 years. And one of the interesting things is that the only way how you can actually sort of establish a connection, and I'm very cautious here to sort of even say the word citation, by looking at geometrical congruence. So I'm fully with you regarding this alternatives to other data types that are, they're much harder to detect. Like photos have this really cool thing, or drawings that, if you depict a ruin, it's either geometrically congruent or it's probably made up. But there is an interesting thing if we think about like what are the sources, and you have, you had this duplication, overlap, and so on coming out of your source material in a sense. But there is another source, which is sort of like the experiment itself, right? And so historically speaking, there's sort of four sources. One is the ruin Number two, and then it already becomes fishy, is other ruins. So if you want to have your Western blot say something, and you take another Western blot and Photoshop it accordingly. And then the third thing is you have it from other documents. So you make up the shape of your picture from some kind of theory, and that's like you get to the, this whole creationism and sort of like making plots show something about vaccinations and whatever. But the most crazy thing is actually the fantasy of people. And so one of the things that I carry around with me since sort of like the first time I saw that is Binet Mandelbrot said, all Financial Times series that are one over F look the same, but sound different. And so the question is, do you think there's other forms of measurement with which we can look at these images, for example, things that are not so easy to spot by humans, like the structure of the noise and the speckles or something like that? Do you do that, or is that sort of something that's sort of too far out because there's a too large set of things one could do theoretically?
Yeah, I'm not sure if I can answer that question really well, but I mean, and I'm not even sure if I completely understand the questions, but I'm good in finding duplications. So that's what I'm good at. But then, for example, when I look at these material science images, which is not my background, I might spot a duplication, but then other people who are really good in that particular technique will spot other problems I would never have found. And sometimes they can teach me to find those problems. And I sort of add a module to my knowledge, it feels like, oh, now I can spot that problem as well. But then there's even other people. When I post these things on social media, sometimes people say, yeah, but it doesn't look correct. Like, this doesn't look like a real experiment. Like, this should have had more background or less background, or this is not the technique they say it is, it's something else. And so other people with more knowledge about certain techniques will spot even more problems in these images. And that is, I see comments on Papier then that I'm like, oh, I would have never spotted that. Like, or this cell line does not have this particular protein. And so why this experiment cannot be true. So there's a lot more knowledge that other scientists will have that could lead to finding more problems. And I'm not sure if that answers your question, because it's maybe a little bit more philosophical, and I'm definitely not good in that. But yeah, that is what I would say. Like, I see a certain problem, other people might spot other problems. And so together, sort of common knowledge, you might find dozens of problems in a paper that I would have only spotted one or two.
Yeah. Mila Oiva.
Yeah, thank you. First of all, thank you very much for your work. It's super important. My background is in cultural history, and I have been studying reuse of text and in different contexts, and also now I'm interested in reuse of different kinds of images in other contexts than science, and of course, within this context that where you are working, it's obviously problematic. While in my studies, it's not necessarily a problem. But I think that overall, it's interesting how repetition is something like that is, it has spread widely in human activities and human culture and so on. But I wanted to ask you, because you mentioned briefly, I'm not sure that I understood correctly, but I think that you mentioned briefly that you think that misconduct and this kind of like, scientific plagiarism and misconduct is increasing. So I just wanted to ask whether you think that that's the case, or whether it is more a question that we are now with the different technologies better able to spot that, or if it's really the case of this kind of like increase of misconduct, what do you think is the reason for that? Thanks.
Sure. Yeah. No, great question. When I did my research on 20,000 papers, I also looked at the amount of problems that I found over the publication years. And I definitely saw I started around 1995, and we saw an increase around 2004, 5-ish. You saw the amount of duplications rise, like the percentage of duplications. So, and of course, there's several reasons. I mean, first of all, papers from 2000 and below or older than 2000 have fewer photos, much fewer photos. If you compare a science paper from 1995, when I did, I was close to finishing my PhD, and compare that to a science paper from now, and by science I mean the journal Science, you will see that journals, these articles are much more complex. So they have many more photos in them. And so there's also a question of the denominator. Like, yeah, we find more duplications, but there's also many more many more photos to look at. Like an old science paper from in the 1990s might only have had one photo. Like, this is the experiment we did, and like one sequencing result or so, and one line graph or so. It's like they were much more simple. So there was not that much to see at. And another factor is that digital photography is a thing that came more used in around the early 2000s. Like when I submitted my first papers around in the 1990s, you had to go to the photographer of the institution who took the photos of your gels, and then you had to make 5 copies and send 5 copies of your printed manuscript with your 5 copies of each of the photos with a paperclip attached, and you send it in a huge envelope to the journal you're submitting the paper to. And it's much harder to cheat because you couldn't just, Photoshop or clone a photo. You had that physical photo. that was all you had. And so the photos didn't look as great as they do now. And we're all used to filters and so. But I think the digital photography, when we are switching to digital cameras, where you didn't take your photos to the photographer anymore. So now it became easier to Photoshop. And this Photoshops from the early 2000s are very clumsy, actually. But now, of course, they're getting better. So they're much harder to spot. So it's sort of a change. I do think the golden age of catching these things is between 2005 and 2000, let's say, 23 or so, because then AI came up. I'll talk about that in the next couple of slides if we still have time, or people still want to hear more a little bit about it. But so it's not just there's an increased pressure to publish. I think we all agree on that. But there's also things like digital photography and the higher complexity of papers that plays a role. So it's really hard to answer a question like that. But I do feel when I did my PhD, I didn't really have the pressure to publish. If I had one paper that was published in my PhD thesis, that was good enough. But now I hear of grad students who say, no, our university requires us to have 4 chapters being published papers. I'm like, I wouldn't have never gotten my PhD with that rule, because I only had 1 or 2 published papers. So I do think there's an increased pressure on everybody. There's also an increased number of papers that are being published. Again, another denominator that you have to take into account. So it's really hard to answer the question, but I do think that most people agree that there's an increased pressure to publish, and thus, at least in absolute numbers, an increased amount of papers that are the results of misconduct.
I think Michael Thomas has the next question. Yes.
Yeah. Can you hear me okay?
Yes, even though you're muted, I can hear you.
Because I'm sitting next to Max.
I need to move the camera so everybody can see him.
Oh yeah, I can actually. Oh yeah, now I see the duplicated things in the blackboard behind you.
Yeah,
yes. Okay, I can hear you.
Okay. So thank you so much. I'm enjoying it a lot, and thank you for the work you're doing. One thing I wanted to mention, I felt guilty at some point because, well, actually I can confirm that your type 1 duplicates can indeed happen just by mistake. I have, in my last paper published like last month, we did have, we have sort of a big picture with 17 or so objects, and they by mistake put two copies instead of one of them, put a copy of another. Luckily the referee noticed that, and of course, I mean, things didn't make sense with the wrong picture. They do think with the right one. Okay, the question I wanted to ask was basically, are you aware of sort of this Russian language project / community called Discernet?
Yes.
Yeah.
That is what Anna Albalkina works on, and
I see.
yeah.
that.
So I don't speak Russian, so I'm not. I'm not. But Anna Albalkina is one of the other sleuths that I've mentioned, and so she's been very active there.
Yeah, because
But like finding the problems, I should say. Yes.
yeah, and so you are just to distinguish and maybe to say a few words to people who are not familiar. They are sort of it is a initiative originating from Russia, and it was initially focused on dissertations, because at least I think probably in some other countries as well. In Russia, there was this problem that essentially every single influential person in any sort of position, be it business or culture or politics or bureaucracy, at some point decided that they want to have sort of a PhD next to their names, and a huge number of these substandard dissertations were produced, and that is. And actually, I wanted to ask: do you see that there is somehow influencing this problem beyond Russia? Do you think that sort of motivation of having just having a PhD to its name, and how big it is well, also there is this argument that part might not be that much of a problem because everybody understands that it is substandard science, and it is just sort of producing fancy letters behind the people's names, and nobody takes it serious.
Yeah, I mean, there was actually when I started working on plagiarism, I soon got into contact with Deborah Weber-Wolf in Germany. She's at the one of the universities in Berlin, but she's retired now. And she worked on a similar project called Vroniplag, which was in Germany, and looking specifically at PhD theses of medical doctors who had to write a thesis but had no interest in doing science, and so were massively plagiarizing each other, and so a very similar project, and there was this whole underground I forgot what it was, like you had to log in somewhere, sort of like a secret Slack, but before Slack existed. This was in 2013-ish or so. And I worked together with them a little while, but their way of working was so thorough. Like every word was weighted, whether or not it was plagiarism, every dash and every umlaut and so I was like, we're not making any progress here. Like they worked for months on one PhD thesis to produce these really detailed graphs on like how many of the pages were plagiarized. So yeah, I worked with them for a while, and then I decided, I just want to move on and wish them well, and I do my own thing, and then I also looked more at images, which I thought were more fun than plagiarism, because you could argue that plagiarism, of the three types of misconduct, plagiarism I feel is the least of my concern, because it doesn't necessarily bring a false narrative into science. And it's bad, of course, because it doesn't give credit to the person who came up with those words or who came up with those ideas. But it doesn't necessarily hurt science as much as falsification or fabrication do, because there you're bringing lies and untruths in the world. So I decided that the duplicated images were, well, first of all, they're more fun because I'm super visual. But second, they bring in a false narrative. So they're hurting science much more than plagiarism. And we can also have that same discussion with AI, because we learn, okay, you cannot plagiarize. We've all learned that. You cannot use those exact words. You have to reword them. And if you reword them and you cite the citation, then it's all good. Then it's not plagiarism. But now, of course, with AI, and this is something I was going to mention on some of the next slides, is it is rewording, right? You ask AI to rewrite a text and it's rewording it, but it still feels like it's cheating because you're plagiarizing another person, just using a machine plagiarizing it. So it's very fast. And if you are a bad actor, you can just rewrite the same paper over and over again and publish that with different authors and sell them to different authors and make a lot of money. But so it sort of changes the whole field, but it's not, again, it doesn't feel it's as bad as making up data sets. That is really much worse, in my opinion, than plagiarism.
Super nice. There's
just
go ahead.
To follow up on this, that in this Russian dissertations business, there is lots of data production, data duplication as well, and there is sort of this. I think the most famous one was guys who took a dissertation about production of chocolate and replaced chocolate with meat everywhere, and sort of white and dark chocolate with beef.
Well, there is white and dark turkey meat, so so that could work.
Yeah, and with all the numbers remaining the same.
Oh.
So we're not a conspiracy podcast, but I think it's sort of like interesting that you bring up Plug via Russia. I'm German, and one of the things where it got famous was actually because there was one very high-profile case which exactly had the PhD being written for him, in essence, due to sort of like they wanted to be even more high-profile as a politician, which was this guy from Karl Theodor zu Guttenberg, who also is sort of like a big landowner in Germany, in northern Bavaria, just saying. And so one of the interesting things about him is that he had to step down as the Secretary of the Exterior, like basically Secretary of State of Germany due to that, or was it defense? I don't know. So he had to step down. He became an elder statesman in the think tank of Henry Kissinger, is a close friend of Sebastian Kurz, who works for Peter Thiel. Both of them recently organized a conference somewhere in the Alps where the current Minister of Economic Affairs of Germany was in her private time, but with sort of like paid by the government. So that was a sort of mini scandal. And she happens to be the life partner, the new life partner of von Gutenberg. And so there's this sort of like weird thing that indeed they fell, like, there was somebody very high profile who fell over their own plagiarism thing. And that has become in Germany so enculturated that now this is the way how to get rid of people. Like there was a Supreme Court justice who was about to be confirmed and she was pro-abortion. And so the super alt-right people, they have now sort of like these Plug equivalents who just, I think 2 or 3 days before she was elected or not elected to the German Supreme Court, Verfassungsgericht, she was actually accused of having plagiarized from a book of her husband. And that was factually the reason she was not elected, even though it had no substance. So this is now used sort of like in that way. And I think it's quite interesting that Gutenberg, for example, also was one of the financiers of an artificial intelligence startup called Augustus Intelligence. And so the Augustus is obviously a reference to Roman emperors, where the key founder was supposedly the Mozart, like a young boy being like highly talented in artificial intelligence. So there is this sort of like thing of harnessing artificial intelligence now in the same way as plagiarism has before, to sort of produce sort of like larger amounts of material. And so one sees a pattern of like, in essence, similarity being sort of like put in front of the chariot, right? And this work becomes actually even more important. The question is, how do we do this with artificial intelligence? Before you go on and answer this question, hopefully, or like...
Well, no, I'm not going to answer.
Tell us how far we are. Sorry, wrong word here. We should talk a little bit about this phase, 2004 to 2023, because I think digital images are like one aspect of it. There is two, like having been, I'm a Adobe Master Collection power user, unfortunately, since Deluxe Paint 2 in 1991. And so I'm a heavy user of the same tools for other reasons. Like, for example, in Renaissance drawings, you often find two kinds of drawings superimposed on each other. In order to read both of them, you need to remove one layer in order to see what's left, because they're different colors or whatever. And so I remember that in 2004, which was around the time I did my PhD, the Photoshop stamp tool, which you can copy one area of an image to another area of an image, became way more comfortable and way better in doing what it does. So that is a really interesting thing. So you could copy the paper structure sort of like into other areas of pictures to make them even more readable. So that's one thing, which I think is quite interesting. So all these techniques to sort of modify images, which you emphasize. But then there's another thing, which is, I think, quite interesting, which is that's about the time when Google Scholar was introduced. Before there was NEC Sightseer, which was used by people in the know, but like Google Scholar was really felt like an explosion. Like people around us, right, started using it even though they'd never used the database before. And I think that is a sort of really important thing pointing to that other aspect, which is you're sort of like looking at the similarities and plagiarism and misconduct in individual papers, but you do so for 10,000 papers and you look at the sort of like summary statistics. But there's this other branch of people who actually sort of like looked at that system in a different manner, which is the citation people who do bibliometrics and stuff like that. And they're the big question, for example, is how much does bullshit proliferate? So I worked for a database company in the 1990s where we did what eventually became a knowledge graph, where we tried to normalize all information. And one of the core reasons was the stratigraphy of the Kerameikos in Athens was used to date excavations all across the Mediterranean, and then somebody found out that the stratigraphy is BS. And then obviously the big question is how many people actually cite that stratigraphy, because that means like all these excavations who cite it probably are problematic. And so the idea was to sort of like light the Christmas tree in the citation network to find these problematic things. And I have a feeling there is like that goes together. So for my question for you in this regard is, if you have your 800 papers, do you see this sort of like, you could say production clubs of this kind of stuff where people copy methods and stuff like that, where you can see, oh, this field is completely infested with sort of fraudsters, while this other field, they have control mechanisms and stuff like that, and we could copy that. So do you see this like social structure behind it in essence?
I don't do that type of work, but other people do. So maybe not exactly in the way you describe, but for example, Guillaume Cabanac, who I already mentioned, one of the sleuths, he does it more on a professional basis. So he has this classifier called Feet of Clay. So if you go to his problematic paper screener, he looks at papers that cite papers that have been retracted. So sort of as a way, because like very often these paper mills don't only like cheat with the data, they also cheat with citations. So they cite each other's work in sort of a citation ring. So it's not exactly what you said, but it is looking at citations and discovering networks of fraudsters, if you will, where, for example, you can see that certain papers are always cited together, where reviewers or authors or paper mills have set up these rings where you always cite one paper from this group of other papers. So like there's editors or there's authors who have these databases. Like if you write a paper, you have to, or you become an author on a paper mill paper, you have to cite at least 5 papers from this set of 300 papers. You can pick and choose whatever, but you have to cite at least 5 papers from them. And that way you can make money, or you'll get a discount on your authorship. So there's these citation rings where people cite each other's work, not because they think it's a relevant paper, but they need to include that paper. Otherwise, they're tossed out of their little group of cheaters. So it's slightly different than what you said, but there's people working on that. That's not me. I'm not very good in, like, you need to have some programming skills and go to OpenAlex to, yeah, that is not something I would be good at, but I just do the images. I'm best in doing that. But other sleuths are looking into those things as well.
The reason I bring it up is because this, there's this sort of Leibnizian sort of idea that all of science is about order, similarity, and relationships in general. And this relationship in general, like how stuff is connected, I think is really hard to tackle historically. Like the material I work with, there is no citations. Like if you look at 10,000 Renaissance drawings, all you get is geometrical congruence. And to reconstruct the network, you need this kind of congruence. And one of the interesting things is that is where your method become really strong and put what they do on steroids, because the citation is cheap, but if papers use the same, say, typography, the same layout algorithms, the same figure panel construction, and so on, all of a sudden this becomes like, the plot thickens in a sense that there is hanging together, right?
Yes. Yeah, and that is, I've written a blog post about the tadpole paper mill, which is a set of 600 papers that we found that all had the same background in the images. They were artificially generated, some primitive form of AI type of images that like gen technology, like what is it, Gen
AI, yeah.
GAN, generational adversarial networks, where you have libraries of Western blot bands and you sort of make a new Western blot bands out of that library, sort of a primitive form of Gen AI. But the fraudsters all use the same background, so that's how we could just recognize, it's again the same background, so this is another paper from that set. But those papers all had very similar structures, like they always had the same type of headings in their materials and methods. They all had a very similar style of writing, and so now you can use AI to find these papers just by their writing style and find more of these papers. So that is sort of the paper that I mentioned earlier from Reese Richardson, published last year in PNAS, looked at that type of analysis, can you find these networks of papers that use each other's images? That's another set of papers that I found, 125 papers that all reused each other's images. And so you can build these networks just looking at images, but other people do that based on citations, based on one particular sentence that you can use as a hook, and then find that sentence is just one little sentence that would not really be detected. It's usually in the figures where you can see they all use the same style and it all has the same source. Yes. So those are paper mills. Yes.
Interesting. That reminds me of art forgery. Like if you have sculptures, then at some point people figured out, if the sculpture is in the earth for 2,000 years, roots grow around it and you have that root pattern on the marble surface. And then obviously the next thing people do is find some fast growing plants and keep their forgeries for 10 years in their garden and then stick them back out. So the question is, do you see that there's a kind of an arms race going on?
Yeah, absolutely. Because we saw these sets of papers that had no background in the Western blots. Then you can see the next generation has all the same background, but like some noise, but it's all the same. And then we saw where they added these artificial little dots and things. Like sometimes you can buy a table that has this distressed look. And so it looks like the table is, 200 years old, but it's just, they have these little metal things that they just bang into the table to make it look like it has a scratch here, but it's all the same scratch. Like if you compare all the tables, they're all different, but they all have the same scratch and they all have the same indentation. So we saw all these papers with the same background stripes and noises. I don't know, it was just very cleverly done. And now we cannot recognize them. So the paper mills are getting better. We could there was a phase where we could catch them, but the fraudsters are getting much smarter, and so we cannot. I think even with AI, of course, especially with AI, they're getting so good. We sort of have a feeling that these images look weird, but we cannot find a smoking gun anymore. So
Seems to me that the one way to win is like forgery being harder than doing actual science.
exactly.
Than to win against Elisabeth Bik.
Well, and yeah, with AI, forgery is getting much easier, right? I could write in theory 5 review papers a day using ChatGPT and get them published. But so I think Gen AI is going to make it much easier to cheat in, on a larger scale, almost undetectable. And I think we could have a discussion on whether an AI written review paper about, I don't know, how good antioxidants are for your health, like some random topic that we don't really need another review paper because there are already millions of review papers about that. But AI can whip out another one in seconds. And, is that bad for science? Or should we just raise the bar on these types of papers? Like, we don't need another review paper on that topic. And especially, like, we need more research papers, and we can have AI write the review papers, of course, but we should not put any emphasis on those anymore. But that's a separate discussion.
Yes, maybe go on with the AI because
Yeah.
we're already deep into our time slot.
Sure. No. Happy to do. So, I mean, I'm not an AI specialist. I'm actually asked nowadays for interviews to say something about AI. I'm like, I don't know much. Like, I'm using it, but I don't really know much about it. But I guess not. That makes me an expert on AI in the eyes of some journalists. So here's my view on AI. It can help us in discovering fraud, but it can, of course, also generate more fraud. So there's a good side and a bad side to it. And of course, there's a whole middle area where I don't really care that much about AI anymore. As in the beginning, like last year, 2 years ago, I thought all AI written articles were bad. But now, as I'm starting to use it more and more, maybe it is, it's new technology. We should embrace it. And I'm using it for help and discovery. So just some things that I think it can help us, like for people for whom English is not their first language, which is probably many of us. It can help us become a better writer, more clear, more sophisticated. And I use ChatGPT where I write a sentence and it makes it much better. And I'm like, yeah, it's still my words, but it just sounds a little bit better. And of course, AI, the machine learning, not the generative AI, but the machine learning type of AI can help us with data analysis. And then there's a lot of discussion, should we use it for peer review? As the amount of papers is increasing, the amount of manuscripts, and while still the same pool of reviewers is supposed to review this growing amount of papers, should we ask one of the LLMs to peer review a paper? And I'm playing with that myself, where I'm not looking at during peer review, because you could argue that if you are a peer reviewer and you get a manuscript and you dump that in ChatGPT or whatever, it's a breach of confidentiality, because now you're actually feeding this into some model that now has that new knowledge. And even though you're supposed to keep the manuscript confidential during peer review. So a lot of journals prohibit the use of LLMs to help with peer review. But other journals are playing with the idea, like, well, we cannot find a peer reviewer for this paper. Let's at least use ChatGPT for a first screen, for example. So this is, I think, a new development where we're still having to form an opinion on what we think. And that opinion is probably rapidly changing. But I'm using it for post-publication peer review. Like, okay, ChatGPT, here's a paper about claiming that, I don't know, COVID vaccines are bad for you. Find 10 flaws in it. And it, within seconds, I have a beautiful pop-peer comment written for me. But the problem, of course, is it makes mistakes. So you have to check everything. So it's still very time-consuming, but it helps me find flaws I might not have found myself. And then we can use AI to do image or like textual language forensics or image forensics, help us find fraudulent papers. So it can do a lot of good, but of course it can generate fake images, fake data sets, plagiarize, like it can rewrite a paper, and low quality review papers. So AI is going to be, and probably is already used on a massive scale by paper mills or by anybody who wants to become an author on a paper and wants to boost their resume. You can just, make a whole industry out of writing very low quality review papers on topics we already have hundreds of review papers about. But you can see that certain authors are taking that route. So just some numbers, like for example, there was a time where ChatGPT really liked the word delve, and you can see the increase of the word delve over time. You see this massive increase around the end of 2022 when ChatGPT became available for a lot of people. And even though the number of papers is on the rise, the number of occurrences of the word delve was even higher. So suggesting that a lot of papers were AI written. And yeah, of course, it produces a lot of slop and low quality papers. Here's a method where AI has been used to detect paper mills, so as a detector. And so this is a paper by the group of Jennifer Burren, University of Sydney, where they looked at papers that had been retracted because they were from a paper mill. And they were very often, these are sets of papers, like the tadpole paper mill I mentioned earlier, that are written sort of on a template. So they change one molecule, they change the cancer, but the papers are not exactly, they don't exactly have the same language, but once you see them, you see they're all sort of following the same structure. And so they used papers that had been retracted for that reason as a learning model to learn a particular machine learning technology to find more papers like that. And so they're finding more and more papers. They screened 2.6 million papers written that had been published about cancer, all kinds of cancer research. So basically all papers that had been ever published about cancer, 2.6 million papers, and found that almost 10% of these papers look like they might have been written by a paper mill. They used the same style of wording just based on the title and the abstract. And we already knew this because we see this massive amount of papers, the tadpole paper mill, I call this the sort of the title bingo generator or whatever. You just use, all the titles have a very similar structure. And so this is what I already wrote a couple of years ago. Once you sort of start to see the pattern, you can almost recognize them by looking at the title alone. And so they all had a very similar title structure. Even though the words were different, you sort of see the rhythm of these titles. They always have some molecule, excuse me, that either inhibits or attenuates or retards, and then cell growth or some process, and then in some cells, and then always have the word by or through or via, and then some word that ends with ing, which apparently is the present particle form. I didn't know that. But once you recognize this title, you can almost see just by looking at the title, that's probably one of the tadpole paper mill papers. So instead of the 600 that we found, just looking at the background of the Western blots that all were the same, now these folks did this research and found there's 10% of whatever 10% of almost 10% of 2.6 million, so 250,000 papers are probably paper mill like. Not just the 600 we found, but many more.
May I ask a question?
Yeah, of course.
I once did I was asked, I had two postdoc advisors. The second one asked me how the first one gets into Science, Nature, and so on so often. And so I did an analysis of the abstracts and titles. And one of the interesting things was the ones that actually make it into Science, Nature, and Cell all had a subject domain, a method, and a criterion of awesomeness in the title. And the ones of that particular group, which was Laszlo Barabasi's group, that didn't make it to Nature, Science, and Cell missed one of these things. Either they were about a method, they were about a subject domain, or and only if they were top tier, they actually had a credible awesomeness criterion. My favorite one is black hole radiation by Stephen Hawking. Obviously the radiation and black hole shouldn't be in the same sentence. And so that kind of thing is sort of something where people actually have this scientific poetry going on where they try to write something like that and be honest about it. Like if the paper is successful, you actually can. So you have two functions how you get to this sort of like conventionalness. One is that you actually make it, and the other one is you just write a conventional title that sort of like fulfills this kind of thing. Obviously, the really good ones tend to be short, but so this is sort of like there's a two-edged sword, right? Where you say on the one hand, the convention is your trigger criteria to at least have a deeper look if this is sort of problematic, while on the other hand, the really good stuff is really conventional because that's when you can actually say a sentence like this, isn't it?
Yeah, no, I agree. And so we don't really know if all these papers are indeed, bad or paper mill writers. But I know as a person for whom English is not my first language, this is how I learned English. You started to see these constructions, like English always starts with, or English, like scientific papers written in English have a particular sentence structures, like however, comma, and then you say something like that is not something that you could find in Dutch. You wouldn't write it like that. And so I learned English by seeing these patterns and starting to write in that same type of structure. So you're absolutely right. Like having the same titles is not just, it could just mean fulfilling criteria. But these are very long titles, by the way. They're not short. but they you can and it might just be, I mean, these were all from hospitals in China, but they all had, it's not just the titles, they all had other things in common. But you could argue, well, maybe somebody a person who for whom Chinese is their first language, it's hard to write in English, and so they might have some language help, like this is how you should structure your title, or this is how you structure your abstract. So you could argue that could be it, but they had many other things in common too. It was more like a sort of a joke, like, oh, this is probably a paper mill production because it has that title, and then we would find other problems in them as well. But we would not use the title structure just by looking at the title. We wouldn't use that as this is a paper mill. But this is sort of what this bioarchive research did. They did look at title and abstract structure to find potential paper mills. But you cannot say on the individual basis, these papers are written based on a template, so that must mean that they're fake. So you cannot make that conclusion. You can just say there's 250,000 papers that are all following a very particular style, and many of which have other problems in the figures, and they have the things in common. They're all from this particular sort of subset of researchers. They're all from unexperienced people working in hospitals in China, like small hospitals that don't normally do research. And you can find other problems. So it's just a language type of analysis that doesn't prove anything, but it just probably hints at a much larger paper mill percentage than we previously thought. But you're absolutely right. It could be just template writing for people for whom English is not their first language.
Cool.
Yeah, but that's of course the risk of doing these large-scale investigations. And then I'm using AI, or sort of, as a way to help me find these duplications. So the two main software tools to help us sleuths find these duplications are ImageTwin and Proofig, and both use AI. But I'm not sure what that means. Like, what part of it is AI? Is it the, more machine learning type of AI where you find patterns, or is it more? There's some intelligence in these programs that know that certain images are expected to be similar, and this becomes very technical. So especially ImageTwin has a lot of that, but both are using AI. So again, I'm not an AI specialist. I'm just using the tools to help me find them. But what most of them have, or both of these have, are large libraries of images. So they have, I think ImageTwin has like 100 million images. from all kinds of papers. So, and Proofig has all the PMC, PubMed Central images, which are all open access. And so they have large libraries. So if I dump a paper into either one of these tools, it will recognize if an image has been published before. Now, it can, of course, recognize itself. So if a paper is in PMC, Proofig, for example, will flag it as, oh, this paper is in PMC, so you can actually check it off. So ImageTwin is a little bit more smart. It will already recognize, okay, this paper is a hit with itself, like all the images match to this particular paper. So it already marks all of these. But you can see that it might find papers that are images that have been published before in other papers or published later. So it not only will find a hit with itself, which it's supposed to do, it will be finding images that have been published before. So sometimes you see that authors are reusing the same control experiment over and over again. Or we've even seen papers that have stolen images, like image plagiarism, from other papers, where they say, oh, this is our experiment, but like actually this paper, you just stole it from another paper and you didn't cite it. So it's sort of plagiarism on image levels. So it's incredibly helpful because it doesn't, like when I scan by eye, I could only recognize problems within a paper. Now with these tools, I'm finding, even in the papers I've screened, I find additional problems, and I'm finding these reused images, which is something that humans are definitely not good in, because you have to see the images together, then you can see it. But I cannot remember, like I said, millions of other papers. So these are the tools. Not only are the sleuths using them, journals are starting to use them as well. Like so Science, Holden Thorpe, the editor-in-chief of Science, has written this editorial where they're telling the world that they're now using Proofig to help screen incoming manuscripts, because you want these to find these problems before they get published, obviously. And more and more journals are using either one of these tools. And also institutions are offering sometimes these tools to their PIs so that the professors can screen outgoing manuscripts before they're being sent out to a journal to screen, especially those papers where you have hundreds and hundreds of image panels. You can screen to avoid making these errors.
Also to help the PI, basically.
Also. Yes.
Interesting.
Well, or to, help the PI not be caught with cheating. You could also argue that, but. So here are some examples of what AI can do. It can create fake images. So Photoshop nowadays, I don't have Photoshop, but Cheshire is one of the sleuths. So, and you can find who he is. So I can say it's a he because that's all public information. But Cheshire is one of the image sleuths. And he wanted to prove in this tweet that you can actually easily generate photos, fake photos yourself, if you have a real photo of real tumors in this case. So in this image on the left, the first, 6 columns are real photos. And then he fed that into Photoshop. There's a generative fill, apparently. And you can say, okay, can you extend this photo a little bit to the right? And it generated an extra row of tumors. So this row is AI generated. I would not have spotted that. I mean, if you tell me, I'm like, maybe you might see that these don't really have a shadow in the same way the other tumors have, but I would have totally glanced over this image and thought it was real. So this row is AI generated, and that is scarily good. And this was already 2 years ago almost. So, and then here's another paper, also a little bit older already, where you can actually build if you feed enough photos to a diffusion model, whatever that means, you can actually generate other photos that are novel, are new, are unique. So ImageTwin or Proofig would not find them, but they're realistic. Like this technology is rapidly improving. I think we've all seen photos and videos online that look very realistic, and as somebody says, well, actually, if you look carefully, 2.3 seconds in, you can see that a lag is disappearing, or you know, some little glitch where you can still catch that errors are being made in these videos. But I think that's rapidly improving, and next year I'm sure we won't find those errors anymore. So if we can do that with videos and photos of humans, or you know, build complete cities that don't exist with this technology, it's very easy, actually relatively easy, to make photos of experiments that didn't happen. So we were at a point where we can no longer detect what is fake or what is real. And maybe instead of trying to prove that something is fake, we need to confirm that something is real, because this is a problem not just in science. It's a problem in journalism, right? I mean, if you're the BBC newsroom and a journalist comes to you with like, oh, there's a big explosion that happened, you know, you used to, the journalist showed a photo and you used to know, okay, yeah, that looks like a real explosion, really happened. But nowadays, anybody can make a fake photo or a fake video, and Twitter is full of them, and generate fake news. And so this is a growing problem, not just in science. And so people have suggested that maybe instead of proving that something is fake, we need to prove that something is real. So have some certificate for provenance and authenticity. And the C2PA is one of those standards that is being developed. So I don't quite know how it works, but it's some sort of blockchain technology type of metadata that will show you that a photo really was taken with that camera or with that scanner licensed to that lab or to that journalist, where you really have some stamp of approval, if you will, that something is real. And this, of course, puts a lot of burden on honest scientists, but I think we have reached that point where we need to prove that something is real, and maybe that's the way forward. But that's just a suggestion that people, you know, people suggested, I guess, that that with AI, that's what we need to do. We need to prove that this photo was really taken in by a scanner in that lab or so.
There is that takes me back to this comment I made with Bernard Mandelbrot, where he said financial time series all look the same, but they all sound different if you listen to the noise on a headphone. And there is in the art world, there's an example where this is already used with art. We have the same problem, right? We don't know what's a real Banksy and what's not. And actually Banksy now sells pieces of art with half a £10 bill. And so his way of sort of like proving that the piece he sells is actually Banksy is by ripping this pound bill into halves, and then the customer gets one half and the other half is stored in the Banksy estate. And there's actually nothing that can sort of like fake the fiber structure of the edge of the £10 bill. It's sort of like, it's really, really hard to fake, and you basically get it on that level. And so I think this is a rather interesting thing, because all of a sudden encryption and, you know, blockchain and all stuff like that would probably come in, where you can trace back all the way to the machine, right?
Yeah, no, I think that's a really good idea. I don't think a lot of scientists would would have a lot of £10 bills to pull that off. But but yeah, I mean it's similar. But that's I really like that idea actually. But there's many. It's sort of like a fingerprint, right? Like we need to work at some way of authenticating that data is real. But I'm a little bit pessimistic actually, because I think it's so easy nowadays to make fake data, and how do we prove that those patients really existed, that you really got IRB permission? Because no longer can we use photos. Photos used to be that, you know, it's hard to fake a photo, or it used to be hard to fake a photo, but now we can make anything. And I think we're all struggling with this. Like if you just look on Twitter, you know, every explosion you see, like, is that really happening right now in Tehran, or is that just a fake video? And a lot of it, I think, is fake nowadays.
One of the things, this is sort of like an interesting thing, that in art history, one of the things we learned is that photography is like painting, and this illusion that a photo actually depicts reality as opposed to, even if it's a sort of like virgin photo, it still has the paper, the way it's lighted. Even simple things like taking a picture of a painting. In the 19th century, they would actually, paint non-reflective furnace over the painting in order to sort of not have reflections when they take a picture. So they would basically change the painting to make it photographable, and which obviously is a nightmare for preservationists. But I think one soothing thing or sort of like disconcerting thing, maybe depending on your perspective, to see is in the 1920s and 30s, the airbrush section in art supply stores, like how much stuff you could actually buy to sort of like deal with photos. And so maybe the time didn't even exist at all at any time that you couldn't sort of like fake stuff. But there was this hurdle that
I know.
Ironically, the distortion of pictures, where you can see, oh, that's shot with a Rolleiflex, or that shot with, or that Western blot was done with that machine because you get that kind of background or whatever, is probably what sort of is the most provenanceable stuff, because really clean pictures are void of these kind of distortions, which actually allow us to tie it down, isn't it?
Yeah, absolutely. I mean, Photoshop has always existed, but it was just so much work to do. I mean, the photos have been retouched, I guess. I'm trying to translate this from Dutch, where like retouched, where photos of, I don't know, queens and kings were always like made look a little bit better or a little bit thinner. People have disappeared from photos just by photoshopping, but it was much, much harder to do in the early 1900s. Now with AI, you could just generate complete fake scenes. So it's always been possible. It's just now so much easier, like within seconds, as opposed to, probably days and days of work. So you can do it en masse. You can do it on a massive scale. You can do it now. And that, I think, is a huge change from the old way that photos were.
I'm super excited. I could listen to you for another 2 hours, but we
I know,
almost hit the
it's
2 hours. You want to close it up, or?
Yeah, I mean, I had much more to say, but I think we had a really, really lovely discussion. So I am worried about AI. We'll see how it goes. Maybe this is just a thing that will resolve itself. But I think we need to slow down science. I think we're producing too many science papers. We need to slow it down a little bit, because science, you cannot force science to be done, in a certain pace. Like, science needs to be slow to be good, and that is sort of what I hope to sort of have the final message. And I think another final message would be, not all science is fake. Like, don't walk away from this talk thinking, oh, let's give up on science, it's all fake. No. Most science is good. We need that to solve the big problems in the world, like climate change, and so we need science to do that. But we need to know which science we can trust, and I think that is a current problem, and we need to work on that. And I hope to contribute to that. So, thank you for inviting me here. It was lovely.
Super, super nice. Thank you very much. As usual, I have to do an announcement what's happening.
Sure.
So we will have the next live session in 2 weeks, which will be Eva Cetinic. And we have another thing, which is next week, where will be no live session, but there will be we don't call it a CUDA Open Lab seminar. We will call it a Cultural Data Analytics Open Lab Special, same acronym. So there's a very interesting thing that coincided. Now our PhD students are sort of like finishing. One of them won the best PhD award at this university last week. And so we're gonna sort of like publish the defense talk, which is not having slides, but moving images dynamically, which is pretty cool. And before we found a recording deep in the archives, which is from 2013, where 3 people, including myself, philosophize about the fact what cultural science, we didn't call it cultural data analytics, could do in 10 years. And now it's 13 years ago, which is sort of embarrassing. We couldn't publish that conversation back then because there's a picture in the background which was not accepted in Science magazine yet, so we didn't publish it. And then it took us another year to publish the paper. So thank you very much, everybody. So I hope you have fun. Follow us on Blue Sky, follow us on LinkedIn. And Elisabeth Bik, we will certainly follow your work, which is deeply inspiring at all times. And yeah, also has a lot of use, not only in biomedical images, but I think in this sort of like whole discussion about like what is similarity, what does it mean, and what can we deduce any kind of meaning from it where we have a sort of like causality in this case. And obviously for those in art history and sort of like the history of biology, we're all interested like how does that actually change over time? So one of the big next things I think we all dream of is sort of like having a lot of your work then being put together and see the system as a whole. And I think that is a good, you know, not diminishing science, but it's actually sort of like standing in front of us as a corpus of something that needs to be more science about. And that I think is a really, really amazing thing that you have achieved this pile of stuff. It's not just exemplifying, but you have, you know, tens of thousands of examples of things where we can actually sort of question science and make it better. Thank you very much.
Bye, everyone.
Bye-bye.